Welcome to 3 Minutes with QIAGEN, where you will get answers to specific questions in 3 minutes. The products shown here are intended for molecular biology applications only. These products are not intended for the diagnosis, prevention, or treatment of a disease. QIAGEN offers complete solutions from sample to insight for performing next-generation sequencing from single cells. The question today is, what are the key factors for a successful NGS from single cells?
The 4 key factors that affect NGS from single cells are amplification of starting material, genomic coverage, amplification bias, and preservation of molecular complexity. The first factor, whole genome amplification, refers to the degree of molar conversion. Protocols used should have a high degree of molar conversion resulting in minimal molecular losses. This is essential to obtain high yields of evenly amplified DNA for downstream steps such as library preparation. The second factor is genomic coverage. Complete uniform genomic coverage is needed to get a good representation of diverse regions within a genome. Gaps due to dropouts can lead to poor data quality and increasing overall sequencing cost. The third factor, amplification bias, refers to the bias due to local differences in GC content, preferential allelic amplifications, and chimeric DNA molecules. It is important to reduce or eliminate bias resulting from the amplification procedure of the starting material. For example, uneven amplification of regions with different GC content would lead to uneven coverage and genomic drop-off or dropouts of such regions. Bias can also be introduced by PCR-based library enrichment that might lead to amplification errors during cycling. The fourth factor is the preservation of molecular complexity. It is essential to use amplification in library preparation methods that maximize the unbiased conversion of unique inserts into library fragments. This will create a highly diverse set of library molecules and lead to more uniquely mapped reads that will give a better representation of diverse regions, and consequently, more uniform sequence coverage. With Repligen single-cell library kits, we try to address these key factors and critical points for successful sequencing. The kits prepare highly complex libraries with complete and accurate genome coverage, even from a single cell, as shown here in the first graph. The streamlined protocol that combines MDA technology with a one-tube library preparation delivers libraries with negligible artifacts, as shown in the table. The Repligen single-cell DNA library kit contains an optimized Phi29 DNA polymerase formulation, which, together with its unique buffer formula, ensures uniform amplification of genomic regions that contain highly variable GC content, as demonstrated in the final graph. Repligen single-cell library kits provide reduced bias and uniform amplification that enables accurate SNP, CNV, and SV analysis. Customers such as Dr. Ludding Song from BGI laud their performance.
For additional information, please listen to our webinar series on single-cell analysis, Sample to Insight Overview.